Some lichens are basidiomycetes, which are what most people think of as mushrooms. And here's a little one, Lichen omphalia umbellifera, growing on a stump. And you can see that it's barely lichenized. In other words, the mushroom doesn't have any algae in it at all. And the algae are all growing on that rotten log. And if you looked closely with a microscope, you would see that the fungal hyphae are weaving in between those algae and attached to some of them and stealing sugars. So we call that loosely lichenized. Oh, for goodness sake, this is going to be terrible. Okay, my mouse keeps falling asleep. I apologize. Another basidiomycete lichen is multiclavula. And you can see both of these basidiomycete lichens usually in the spring when it's wet. So maybe now, or maybe it's a little too late this year to see these two basidiomycete lichens. The rest of the year, you don't know that they're a lichen at all because it's just the green covering on the soil or the log. But most lichens are ascomycetes. So let's take a cross section through a lichen and just see what it looks like in there. There's an upper cortex where the cells are small and usually roundish and stuck together very tightly. Then there's the medulla, which is this whole area here. The upper medulla has the photobiont in it, in this case, a green alga. And the lower medulla has no algae in it at all. It's just fungal hyphae. And then there's a lower cortex, again, made out of small cells of fungus. Here's a different cross section just to show you that lichens are different structurally inside. Again, here's a thick cortex. Here's the medulla that's much airier than the last one I showed you. And there's some single cells of algae in the upper medulla. And then there's a lower cortex. Some more terms we need to talk about are the three artificial groups of lichens that we've just sort of grouped them in this way to make it easier for people to talk about. And the three artificial groups are foliose lichens, fruticose lichens, and crustose lichens. So I'll show you a few foliose lichens. Here's Loberia pulmonaria, that I'm pretty sure you're all familiar with, and Xanthoparmelia growing on a rock. Another one that you see less often, but it's pretty common in the Coast Range, is Pseudocyphalaria hawaiiensis. And it's just so beautiful with these golden ceridia, which we will talk about later. Another group within the foliose lichens is the jelly lichens or gelatinous lichens. And what we're looking here is not at the moss, but at this dark brown thing that's actually really hard to see when you're out walking around because it's so dark. So when you're looking for this kind of lichen, you actually have to look specifically for dark lichens. They're called jelly lichens because when they are dry, they're all hard and crusted and thin, but when they're wet, they're jelly-like and puff up a little bit. So that would still be considered a foliose lichen, but the interior looks a lot different. The upper cortex is usually only one cell thick on a jelly lichen, and the lower cortex also one cell thick. And then the medulla, instead of being separated into two parts with the photobiont on the top, is all the same all the way through with fungal hyphae wrapping around strings of cyanobacteria. Another really important thing to remember is that lichens are named for the fungal partner, which is unique. In other words, each fungus making a lichen is a different species. The photobiont is often shared by many lichens. For instance, in the Willamette Valley here, if you look at the lichens growing on oak trees, almost all of them have the exact same photobiont. So it's clear that we wouldn't be able to name the lichen after the photobiont. On top of that, some lichens have more than one photobiont. And here's an example of that, Peltigra leucophlebia. It's bright green lichen when it's wet, and you can tell that it has green algae as its main photobiont. But if you look carefully, you'll see little black dots, and these are cephalodia. In other words, a little tiny packet of cyanobacteria encased in some fungal hyphae, and they're in this lichen just sitting on top of the thallus. So this thallus is getting the best of both worlds by fertilizing the soil underneath it with from the cyanobacteria fixing nitrogen. And Peltigras in general live on bare soil, usually recently disturbed. And then a shocking new discovery was made a few years ago by Tony Sperbilli. He figured out that a lot of lichens have another fungus that is always associated with them. And this image shows that he fluorescent stained the two different fungi. The green is a basidiomycete yeast, and the blue is the fungus, the ascomycete fungus that we already knew about. The red in the picture is algal chlorophyll, stained red. So nobody knew about this third fungal partner, and it's not present in every lichen, but it is present in quite a few lichens. And it turns out that this basidiomycete yeast might be at least partially responsible for making lichen acids, which are unique to lichens. They're just chemicals that the lichen makes. And these lichen acids turn out to be very useful to the lichen in different ways. Some of them act as very good sunscreens. Others are antibiotic chemicals. And there's several other things that they can be, but we won't cover that. So back to our three main groups of lichens. This is this is the second group of fruticose lichen. Fruticose lichens are bush-like or long. And you'll notice that fruit isn't spelled like apples and oranges. It's F-R-U-T, which is the Latin root, means bush. This is Usnea longissima, and you'll see it around near creeks and rivers in Oregon. A similar looking lichen, but from when you're far away, looks very similar, is the fruticose lichen Ramellina menziesii. But if you look at it closely, you'll see that it's made of little nets. And so it's called the fishnet lichen. And the neat thing about this lichen is if you look carefully at this picture, you can see there are tiny nets. Are you guys, August, are you guys able to see my arrow?
Yep, we can see that.
Oh, good, okay. So it's made of tiny nets like these ones at the ends that grow in every dimension as the lichen ages, so that these tiny nets could become something this big, or even something big enough that this was part of one of the nets at one point, but finally broke. Really beautiful lichen, and it's the state lichen of California. Other fruticose lichens include the cladonias, which are pixie cups, or these British soldiers with little red apothecia at the top. And bushy lichens like our beautiful Letharia columbiana. which grows on the east side of the Cascades, more often on the east side of the Cascades. And the third major group of lichens is the crustose lichens. And in crustose lichens, the thallus is not separable from the substrate. There is no lower cortex on a crustose lichen. So some foliose lichens are tiny and look like they might be crustose lichens, but if you can see a lower cortex, it's not a crustose lichen. Crustose lichens cover the trees out here so much that you often can't see the bark at all. And a good example of this is if you're driving out to the coast and you see the alder trees along creeks, their bark is pretty much white. And if you look closely at them, you'll see that that white is all lichen, and the actual bark is dark gray. Here's another lichen taken in Alaska, crustose lichen. And this is a common lichen around here called Ichmedophylla ericitorum. And this one is a great picture because on the edge of the lichen, as it's growing radially out this way, you'll see that it's releasing a lichen acid that's killing the moss ahead of it, so that that moss can't overgrow this crustose lichen, which isn't able to cover back up if something gets on top of it. And here's the crustose lichen pertusaria, something or other. So let's briefly talk about reproduction. How do lichens spread? Well, around here, the main way they spread is by ceridia, which are asexual reproductive propagules. And you can see them here on the pseudocyphalaria as little patches of bright yellow ceridia. Each ceridium is made of fungal hyphae with some algae inside, just a tiny little bundle, and it can blow or wash in the rain or get stuck to a bird's foot or get eaten by a slug, and then spread along a branch or farther than that. The same goes for these little patches of Ceralia. They're out at the tip of the hypogymnia, and they're in each of these patches is hundreds of little ceridia. Ceridia are very, very light, and so they can blow pretty easily and pretty far. Another method of asexual reproduction is isidia, and isidia are basically the same thing as ceridia, except that they have a cortex on top of them. So the cortex protects the algae inside so that possibly it wouldn't dry up as fast or get burnt by the sun. But the cortex adds weight to that propagule and makes it so it can't spread quite as far as easily as a ceridium. A third way of asexual reproduction is just by breaking a part of the thallus off, and that piece can land and grow again into a new lichen. In Loberia oregana, it has made specific little lobules that break off really easily and can spread and land at the right microhabitat and grow. Again, this is even heavier than an isidium and probably doesn't spread as far. And Loberia oregana grows in old growth forest and doesn't really spread very well out into new forests. So it and many other rare lichens have this problem of the propagules not being able to spread very, very far. They're poorly designed for spreading where we're logging a lot and changing the forest constantly. And then there's sexual reproduction, which creates apothecia. I'm not going to talk about this a lot, but in in an apothecium. Here's an example of an apothecium on a Lobaria pulmonaria. And here, and then these down here. The red is the fruiting surface where the spores will be underneath there. The spores will be produced, and around the edge is a rim. That's just the lichen. It can have in it the algae and the fungus, but the fruiting surface is only fungal. And here's an example of apothecia on that jelly lichen. So in that fruiting surface there, small sacs form called asci, and in those asci are usually 8 spores. And the spores, being completely fungal matter, then are pushed out into the air and blow away and land somewhere, and they have to find an alga or a photobiont before they can grow into a new lichen. So that's a big mystery about how that happens. And I think probably one of the most common ways for a spore to find a new a photobiont of the right kind is for it to land on another lichen that happens to have the right photobiont in it. There are 2 genera of lichens that have managed to put algae inside of that fruiting area that's usually only fungal. And so when the spores emerge, sometimes an algal cell attaches to it as the spore leaves. So that, like, those lichens have figured out a good system there. And here's a cross section of an apothecium. You can ignore this pointer. Here's the rim with algae in it and fungal matter. And this is all just fungi, fungal hyphae, forming little sacs in here. And here are the spores that were in a sac and are now being released. Now I want to change tack and talk a little bit about how lichens are so specific about their habitats. And this sort of introduces the part where I'm going to be telling some stories about lichens that I've known. On a broad scale, lichens can grow, some lichens grow on rock. And what kind of rock do they grow on? They might be very specific for limestone only, or they might never grow on limestone, only on other kinds of rock. They might grow only on conifers or only on deciduous trees. Some grow on both. The amount of precipitation makes a lot of difference to them. Is it only in the summer? I mean, only in the winter, or does it go all year round? Or if there's a dry season, is there fog at that place? The amount of light makes a big difference. For instance, if you're along the edge of a forest or along a trail or a road, that might be a completely different habitat than inside the forest nearby. The elevation obviously makes a big difference because of the temperature differentiation with different elevations. And the pH of the soil, of course, is important to soil lichens. On a microhabitat scale, bark texture makes a big difference. Is the bark corky, or is it absorbent of water, or is it hard and doesn't allow water to come in at all? If it's growing on wood, is the wood hard, or is it soft and hold water? Where on the bowl might this lichen want to grow? Down near the ground might be drier. I mean, moister. Up higher on the bowl might be drier. Location on the branch. My thesis was all about where on the branch the lichens begin. Some start way out on the twigs that are out in full sun, even when the leaves are on the trees. Others can't get a hold until the branch is old and shaded all the time. The aspect of the rock a lichen is on might make a big difference, or it might need to be in an underhang on a rock. And then soil texture, if it's a soil lichen, soil texture is very important. So when I'm out looking for lichens, I'm constantly being reminded that I need to look in all the different kinds of microhabitats where a lichen might live. So I'd like to tell a couple stories about some lichens I've known and what I've learned from them about microhabitat. Here I am at Hovenweep National Monument in Utah, and I was looking at this beautiful ancient wall, and there on the top of it is bright orange lichen. So why is that lichen growing there, not over here? This area is obviously a raptor perch. The birds sit there and they poop, and they bring in nitrogen, and they lower the or raise the pH. So closer to home, I was surveying, I was leading a field trip for the Forest Service. We had just had a workshop on rare lichens and mosses. So we went up to Quaking Aspen Bog to see if we could find some of them. And as I was entering the bog, I noticed this bright orange tree. And I know it's a little hard to see in this picture, but you can see along the bole, it's orange, and some of these twigs are bright orange. And at first I thought it was flagging, but I walked closer and saw that it was Xanthoria, the same bright orange lichen that I saw at Hovenweep, and just covering parts of this tree. And so I thought, well, it's probably a bird perch. Well, since we were on a field trip trying to find rare things, I thought about a lichen I know that grows only on bird perches way up on mountainous ridges on small Krumholtz trees up on mountain ridges. And it grows on trees where birds perch. So I said, okay, let's see if it happens to be on this tree. And somebody got on somebody else's shoulders and reached up to the very top of this tree, way up here, and took some pictures by holding his camera up above his head. And there's the orange Xanthoria. And this is what he found. If you see, you can barely see it in this picture, but there's a stalk that the arrow is pointing to, and there is a black apothecium on the tip of the stalk. And in another picture, you got this huge thallus of this Thalerna dissimilis, a quite rare lichen in the Pacific Northwest, growing there in the middle of the bog, not up on a mountainous ridge. And so actually this microhabitat, or this habitat, is not necessarily mountainous ridges. The habitat is tree perches, raptor perches on the tops of trees. And I suspect that if we looked all around that bog, up at the tops of all those trees, which we can't do because they're really tall, we would have found some more thalli of Thalerna dissimilis. The raptors sit there so that they can look down on the bog and catch small mammals. Well, continuing to talk about Xanthoria a little bit more, another place that you'll see just trees or branches covered with bright orange Xanthoria is along the I-5 corridor. Xanthoria, as I said, it likes high nitrogen and sometimes high pH as well. So along I-5 corridor, there's a lot of dust being blown up all the time by cars. And also a lot of times when you see this, it's also near an agricultural field where the dust is stirred up by plowing. So that sort of explains our orange trees. But in this case, it's even more interesting. Here's Xanthoria parietina, which is, this is a twig from that previous slide. And Xanthoria parietina is an invasive species that came, I believe, from the East Coast, and it's being spread by nursery stock. And the best place to find it is by looking at in a new housing development and looking at the trees they've planted along the street in the housing development. And often it has this species growing on it. It's invasive, but it's hard to know if it's actually harming other species or just filling in spaces on the bark. The next story I'd like to tell is a wonderful story about Mount Pisgah Arboretum's silo here on the old barn. If you look really carefully in this picture, you can see that down in this area of the silo, it's slightly orange. And also notice that there are vertical pieces of wood going up all the way around. Well, if you get close up to that and look in between the two verticals, you will see that over on the left-hand side, on every section on the north side of this silo, on the left-hand side is yellow. And that's because this lichen, Chrysothrix, needs something that's there and isn't over here. I'm not sure if it's shadier or if it's a little moisture or what. So it's always over on the left-hand side of every single section of that silo on the north side. The same lichen is also found on Douglas fir trees, and here you see it on the north side of a Doug fir, out on the outer part of the bark, not nestled in the cracks. But if you look at this section again, there's something that we didn't notice before. There is Cladonia here, one of the fruticose lichens, but I'm talking about this over here. This sort of dark brown thallus with some black speckles on it. So let's zoom in. And you'll see the beautiful little pin lichen, Chenotheca phaeocephala. And if you go to Mount Pisgah and you have a hand lens around your neck, you can find this by looking at the right-hand side of each section between the verticals. Pin lichens are so called because they look like a pin stuck in the piece of wood. And the pin is all fungal material. It's an apothecium that has a stalk and a little cup, and in that cup is tons and tons of spores that have already been ejected from the asci and that are inside the apothecium. These ones are also especially beautiful because they have this yellow dust on the outside of them. That's not spores, that's just the coloring of this beautiful lichen. And since I've started talking about pin lichens, I'll just show you a few more that we have in the Willamette Valley area. Here is a sclerophora, and I especially like this one because it has yellow spores sometimes and purple spores sometimes. It might not be the actual spore, it just might be the coloring on the outside, a sort of dust on the outside. These pins are about 1 millimeter tall, and the two common species here, one grows at the base of maple trees only, and the other grows on old oak trees, usually on a bowl that's slanted where it doesn't get wet. Another pin lichen in this area grows on Douglas fir and cedar trees, and here are the pins, here, and then this one has broken off and this one has broken off. I love this lichen because it has a sort of a nondescript thallus, but along the edge of it, it's forming a lichen acid that is brilliant orange. And if you put KOH on that part of the thallus, it'll turn bright purple. So that's Chenotheca ferruginea, named after the rusty color. And another common pin lichen in the Willamette Valley is Chenotheca furfuracea. Its habitat is in between the roots of a Doug fir tree on the downslope side, in sort of a little shaded, moist spot maybe. It can grow on rocks, on soil, or even on rootlets in that area. And this is what it looks like. Again, the pins are about 1 millimeter or 2 tall. And last, I'd like to tell you about a really special pin. I was surveying up in Washington at a special protected area owned by the Columbia Land Trust in an area with ancient oak trees. And you can tell they're ancient because the bark here is so deeply creviced, furrowed. It's just beautiful. And I noticed some black spots on a white thallus. So zooming in, we found Calicium quercinum. Calicium quercinum is one of the rarest lichens in the Pacific Northwest. There are only 2 sites known of this species, and I don't believe that that is because we haven't looked for it. It has such a specific habitat, which is old oak trees. And I've looked at many old oak trees and not found it. The second site, besides this Columbia River Trust spot, is on the OSU campus, where it grew on a tree along, I think it's 30th Avenue, where there was a stand of nice, big old oaks. And unfortunately, the one that had Calicium quercinum on it fell over. So some of Bruce McCune's student, Bruce is a professor at OSU who studies lichens. Some of his students tried transplanting chunks of bark onto another tree, but I don't think that was successful. So I think that we're back to just one site in all of the Pacific Northwest for this unique lichen. So I started talking about oak trees, and that takes my mind to some really beautiful small lichens that you probably haven't seen because they're small, so I'll show them to you. The first one is Normandina pulchella, and pulchella, as you know, means beautiful. And here is this beautiful little tiny thallus, about probably 1 millimeter across, sometimes bigger than that. And it has its edges rolled in. Oops, my arrow disappeared. The edges are rolled in like an ear, kind of. And it just grows there on mosses or among mosses on bark. And liverworts, obviously, because here's a liverwort. That's Normandina. And here's another cute one, Waynia californica. Unfortunately, this picture shows it sort of being the wrong color because this is from a photograph of a collection, an old collection. And when it's fresh, it's green, like similar to the other lichens we see around here. It forms Soralia, pockets of Soridea in these sort of swollen places, and then it opens up and the Soridea can fly out and land somewhere else. This lichen, we hardly knew it was here until I started surveying and looking really closely at oak trees, especially in Southern Oregon. And then there's Dendriscocolon. Dendriscocolon is a really hard to see lichen because it's brown. It's like one of those jelly lichens, and you probably wouldn't even notice it on a tree unless you were really looking for it. It has a cyanobacterial photobiont, and here it is under a compound scope, and you can see the blue-green, the cyanobacteria in there. And that cyanobacterium is Gleocapsa, a different one than many of our cyanobacterial lichens have. And you might be wondering why I have Dendriscocolon in quotation marks. And that's because Dendriscocolon was thought to be a species, a unique species, for a long time. People knew that it grew often on other lichens, big foliose lichens, but it still looks so different that they were sure it was a different lichen. But using DNA sequencing, people have found out that it's the same fungal partner as some of these large loberias, not this particular Loberia pulmonaria, but other Loberia species. And while the loberias that as we know it, has a green alga photobiont and then a small pockets of a cyanobacterial photobiont inside, we usually don't see any of those cyanobacteria. But when dendriscocolon grows out of it, its photobiont has made the fungus change the shape of the lichen. So it's really spectacular. And by doing this, somehow dendriscocolon has increased its range by hundreds of miles, because the closest Loberia that is the same species as the dendriscocolon that we have here grows in Alaska and not farther south, maybe a little into British Columbia. So by changing to a cyanobacterial photobiont, the lichen has extended its range. And that's just remarkable. Fungi are so cool and so smart, especially when they're lichenized. I think this is the last little lichen I have to show you on a oak tree. And this one is called Placidium fingens. And it's this flat little lichen that's growing on top of another thing here, which is a Cytinium, a jelly lichen. And it only grows on top of those Cytiniums on oak trees. That's the only place it grows. While most Placidium species are soil growers, but on the soil, there are often other Cytinium species nearby. So I'm kind of thinking that this might be a very loose association between the jelly lichen and this Placidium fingens. You'll notice that this has black dots on it, and these are perithecia. They're like apothecia buried inside of the thallus. And here they are. The apothecium has just folded up into an urn-shaped thing, and the spores are produced in asci here and released through a little hole out. Okay. So the last story I'd like to tell you is the story of Faliscum, and I'll speed up a little bit because I see it's getting late. My job at Oregon State University Herbarium is to clean up the herbarium and try and identify the things that aren't identified. So I found this packet, which is on the kind of label that Bill Dennison used to use. So I kind of suspect that he, either a student gave it to him or he found it himself. And it said Faliscum on it, and then it had Hoskins, Oregon, and the date, and that's all. And I said, well, I'll try and identify that. I've never identified a Faliscum before. And looking at the thallus, I see it's a tiny, tiny lichen, and it's growing on very red soil. It also had apothecia on it, which was wonderful because I could look at the spores and see that they had 2 cells. I got, I couldn't figure out what it was. I got Bruce McCune's help on this. And I looked under a microscope and I saw that it had, the cortex is made out of jigsaw puzzle shaped pieces, which is really unusual. I only know of a couple other lichens that have cortex cells shaped like that. So that was sort of a hint that it might be something interesting. And one of the genera that has those cells is Polychidium. So I thought it was probably a Polychidium. But not only did the spores not match up properly, but the photobiont was the wrong photobiont for the kinds of Polychidium that have puzzle-shaped cells. So Bruce and I decided we better sequence it. And to sequence it, we would have to go find some new thalli, because the ones from 1969 would be too old. The original label said it was near Hoskins, and Bruce knew from talking to some of Bill Dennison's sons that they often went for hikes along an old railroad that went between Hoskins and Valzette's. So Bruce and I drove out there. Oh, here's Corvallis and here's Albany. So it's sort of near Corvallis. We drove out there and we drove along a road that's now where the railroad grade was. There's no more railroad. And we drove along that road and we looked for patches of soil that didn't have too much leaf litter on it and didn't have too much vegetation, but was red. And we finally found a place, oh shoot, we found a place like that. I don't think, oh yeah, I can go back. And collected some stuff off the soil, tiny stuff. And when we got home, it turned out we had collected the right thing. So we sequenced it, and the sequences told us that it was closely related to Peltigera. Peltigera is a big foliose lichen. It's maybe a centimeter or two wide and 5 centimeters long sometimes, and very obvious on all over the place. And it does have a cyanobacterial photobiont. The other thing, the other species that the sequence came out near was Solarina species. This is Solarina crocea, and it grows up in the mountains in wet spots with a bright orange underside. And another species looks more like our little thing, sort of tiny little dark lobes with big apothecia nestled in there. And we didn't believe the sequencing. We thought something had gone wrong, so we tried again. And no, it really is in the same family as these two lichens. And it turns out it's a completely new genus, and the only species that we know of so far is this species. So we named it Sinuosella after the sinuous cell walls here. And the species Dennisonii is after Bill Dennison. So that ends my talk tonight, but I'd like to just, show you a couple references if you're interested in learning more about lichens. Macrolichens of the Pacific Northwest is a wonderful book written by Bruce McCune and Linda Geiser. Be sure to get the second edition. If you like to work online, there's a new website, Common Macrolichens of the Pacific Northwest by Bruce McCune and Sunya Yang. And you can find that at the Northwest Lichenologist website under Literature and Keys. Here's the Northwest Lichenologist website, and you can just Google Northwest lichens or something like that, and you'll get there. We have a forum where you can ask questions. You can post photos and ask questions about them. We have news of talks and things happening. Resources are lots of papers that are recent papers and help you identify things. We have a certification program, lichen certification for the west side of the Pacific Northwest, where lichenologists can get certified that they are competent in collecting and identifying lichens, and especially the rare lichens that we have. We also have a small store with t-shirts and other things. So thank you very much for coming and listening. And I'll stop my share. Maybe.
Thank you, Daphne.
You're welcome.
And I encourage everyone to unmute themselves. Let's do a quick round of applause before jumping into questions.
I'm trying to stop.